Hi, so my name is Timothy James, and I'm a vertebrate paleontologist living right here in Southern California. And today I'm going to teach you a little bit about the age of mammals, and more specifically, elephants that lived during the age of mammals. Now you may be familiar with woolly mammoths, or American mastodons, or even the modern day proboscideans we know and love, like the Asian elephant or the African elephant. But today I'm going to be talking about one of their early relatives, and that is, of course, the gomphotherium productum. Now, this animal is significant because it had a geological lifespan for over 7 million years. Now, what does that mean? What is a geological lifespan? So a geological lifespan is just how paleontologists look at how successful an animal is. For example, the normal geological lifespan of a successful mammal species is 2 to 3 million years. This individual subsisted for over 7 million years. And just combine that with what we know about human evolution, with the earliest Homo sapiens coming around 300,000 years ago. So that just goes to show just how successful this early species of elephant was, if it lasted for 7 million years. And that's just in one certain locality. Now you may ask, when did it persist? And it persisted from 14 to 7 million years ago. And that's a time that we call the Middle Miocene period. So it's a little bit before the Ice Age, when the Earth was a little bit more of a grassland savanna type environment. And we look at specific individuals coming from the Espanola Basin of New Mexico. Now that is just outside of Santa Fe, if you know where that is, and the Espanola Basin actually holds some of the most exquisitely preserved individuals of this species ever discovered in the Americas. Now, why does this matter? It's because in paleontology, what we're trying to do is we're trying to construct extinct ecosystems from just a few small pieces. That's like trying to reconstruct the Amazon rainforest from a few pebbles and rocks and assorted bones and a piece of dirt, right? It's not an easy thing to do. So that's why our sample size is everything. So a majority of what we're trying to do with this research was expand that sample size to determine whether or not there's a dwarf species of Gomphothere represented within Gomphotherium productum. And one of the ways to do that is, of course, by paleontology fieldwork. And so what we do is we go out into the Badlands, these areas where you see, layers of time eroding out with no plants. Think about it as a chapter in a book that you open up. And we walk around till we find little bone pieces on the ground. And once we find those bone pieces, we try to go and see where they're eroding out of. And once we find where a bone is eroding out of, that gives us a chance to get what we are looking for, and that's an associated site. Because an associated site is not just one remain, one part of an animal, just one bone, but it's a representation of that animal's skeleton. And once we have that representation, then we're able to reconstruct much more features about that animal, including diet and even the ecology of the environment it lived in. And what we did here is excavate a Gomphotherium productum skull in the middle Chimeta member of the Prohaje Formation, named after an indigenous group in the area. And what we found was a exquisitely preserved Gomphotherium skull, known as Gompy. And it's seen here, our process of actually collecting this so that we could study it and actually add it to our results. And so what we did first was isolate where the skull was and trench around the skeleton in order to get underneath and preserve the specimen with as much matrix or dirt around it as possible. And so what we do is we put plaster of Paris once we've trenched around it. And if you've ever had a cast, if you've ever broken your arm, it's the same general concept. We get burlap and plaster of Paris, and we create a cast around the skull that allows us to transport it with the dirt and the matrix around it safely into the lab. And sometimes it's really difficult to get these multi-100-pound blocks out of the desert. So we have to use unconventional means. One of the ways that we got this piece out was actually putting it on a car hood and then grabbing ropes and actually pulling it as we go to get it back to the museum in one piece. Now, once we got back to the museum, of course, it's covered in all this matrix, millions of years of accumulation. But we can't just blow through that matrix because it contains valuable pieces of the past. things like fossil pollen that can tell us what kind of plants and what kind of flowers are existing, things like seeds, and even insect remains, freshwater snails, etc. All of these things can be found in the matrix surrounding the bones. So what we were doing is using very non-invasive methods to actually prepare the fossil and get all the material off of it. So what we use is dental picks, kind of like the same thing. If you ever go to the dentist, we use the same tools. And so we'll use dental picks, and we'll also use pneumatic tools similar to a dentist, but instead of a little Dremel, we actually use an air scribe. And so think of an air scribe as a a miniature little jackhammer that uses the air pressure and vibrations in the matrix to separate the matrix from the fossil. Anyway, once we've completed this process, which is an arduous and difficult, long process, for every 1 hour you spend in the lab, you spend 1 hour you spend in the field, you spend 5 to 10 hours in the lab. It takes a long time. But once we've prepared it, then we can do things like body mass regressions that allow us to determine how big this animal was, which is crucial because we're trying to see if there's a dwarf species represented within this sample. And so once it's prepared, what I was able to do is actually do measurements of the palates of these different proboscideans and compare them with the different measurements in the collections. And we were also able to do some body mass regressions to try to see what the range of individuals represented within the Espanola Basin specimens showed. Now, unfortunately, sometimes as science goes, we didn't have any conclusive results, but that's not necessarily because the data's not there, it's due to the lack of sample size. So in the future, this project will continue on, and I will be sampling gomphotherium productum specimens from the Frick collection at the American Museum of Natural History, from the University of California, Berkeley Museum of Paleontology, as well as the tar pits and any other museum in the Western United States that may contain specimens from this locality, to thus understand what's going on. And I'd just like to thank the other people who were involved in this research, my co-authors. That's Gary Morgan, the curator of paleontology at the New Mexico Museum of Natural History, and Jonathan Keller, a PhD student in the Smith Paleoecology Lab at the University of New Mexico. This was a joint project between the University of New Mexico, the New Mexico Museum of Natural History, and the Smith Paleoecology Lab in the biology department at the University of New Mexico. Thank you very much for sitting through my lecture. Do you have any questions?
I actually do. I was wondering what made you think that this was a dwarf species, or like what if it was just a baby of like a child of a mammoth, or like a different type of gomphotherium, I guess.
That's a great question. So one of the first things we're trying to rule out when we look at different fossil specimens is determining where the individual fits into the ontogeny. Now, ontogeny is just a really fancy way of saying the lifespan of an animal. So we were actually lucky enough to actually sample juvenile gomphothere palates in order to create a control. And you can see here that we had 2 individuals that we sampled. Now, we're always looking for more juvenile gomphotheres to study, but we were able to. The other difference between the juveniles and the adults that we're looking at is juveniles actually have an extra tooth in their mouth. Similar to how we have baby teeth, they have baby teeth as well, except those baby teeth are actually known as milk teeth. So they have an extra tooth on their lower jaw and their upper jaw that allows them to suckle their mother's teat. And so that's one of the things that differentiates them from a potential dwarf species.
So you said that they lasted up to 7 million years. My question is, what eventually ended up putting them extinct, or are they still around today?
So that's a great question. So these animals are what I would call a victim of their own success. They, gomphotheres ended up branching out and becoming so successful that by the time Gomphotherium productum was reaching the end of its geological lifespan, it had multiple other species of gomphotheres that had evolved from it, living in its same environment. So you had rhynchotheres and Cuviornis, and it was also competing with other proboscidean branches like the mastodon and the mammoths. And because of this, its niche really got eaten up. Now, just like actors are typecasted, animals have their own niche that they look for in order to survive and persist. And essentially what happened is this animal's market share in the environment continued to get eaten up by all the animals that had evolved from it. And so because of that, it went extinct. And of course, there's climate reasons and a whole plethora of factors that plays into the extinction of any animal. It's always accumulation of causes, never just one singular event. But its ancestors, its descendants, specifically the Cuviornis, a late gomphotherium, actually persisted way into the Holocene period. And there's fossils from Cuviornis that may have been represented with butcher sites in northern Mexico in the Holocene period. So it really just goes to show just how successful this animal is. And it also represents a very basal proboscidean that is a distant relative of modern day elephants. So its genus is still surviving today, and it is one of the last surviving examples of Pleistocene megafauna on Earth.
That's so cool.
Well, if there's no more questions, I'd like to thank everybody for coming to my talk today. And yeah, I hope this has inspired you to learn a little bit more about the Ice Age history and the age of mammals in the places you live. Thanks for letting me share about New Mexico's paleoecology.